Hi, I'm Dr. Ben Neuman, Texas A&M University, and this is my little time capsule. And we'll see if it ever gets opened, won't we? All right. So we've heard a lot about SARS-CoV-2 at this conference, and MERS and a couple of the human ones. I wanted to use this time to reflect on some of the other viruses and show you some really neat things we found, particularly in the invertebrate, Nido and Nido-like viruses. So this is the Nidovirales you know and love over on the left, plus about 50% more taxonomy. Yeah, perfect, right? All right, we'll start out just by showing this. This is a picture of the variant distribution on the Texas A&M campus over the last 6 months. And what you see here is that we have been replaced from a very diverse ecosystem to a monoculture of the orange one here, which is the B117 variant. And this has happened as the case numbers have been falling down. So it looks as though selective conditions and the rise of this virus go hand in hand. Yeah. But this sort of complex interplay that's playing out everywhere and that we're all paying attention to is also happening among the invertebrate nidoviruses all the time. And it's important to keep that in mind, that there's a world that's bigger, and spillover from that world is how we get, bad new things in vertebrate animals. All right. These are most of the viruses that we found, and I just want to use this as an overview. Let's show you some of these groups. At the top in green, those are all the Proto-Nidovirales. So this is the sister group to the Nidovirales. And if you look at the rooting, we used 5 different caliciviruses as our root, and that appears to be a slightly earlier branch point than the actual Nidovirales. So you can look at them as either ancestral to the Nidovirales or possibly as a sister group, because this is just a very quick and easy phylogeny, and there are better ways to do this that might be able to tease out sort of the precision on that particular thing. So take that bit with a grain of salt, but in general, yeah, some really neat viruses, all in invertebrates. In the center, we have our only vertebrate viruses that I'm going to show you, which are some of the lito-like viruses. It's going to be similar to that sister group, the microhyla litovirus, which was found in a little frog. And the rest of these are all in fish. So these are the earliest branches inside the Coronaviridae. And look as though they are the only ancestors that we know right now of the actual original coronavirus. The other interesting thing here, I think, or to keep in mind, is that we have very incomplete sequence coverage on all of these branches. And so this is kind of like trying to put a forest back together from a box of toothpicks. You'll have some of them will line up, but there's a lot that is missing. So I would expect a lot more branches to be filled out on these particular trees in the coming years. But the neat thing is the Proto-Nidovirales in particular has 20 new sequences in it, and those are in 3 suborders as far as we can tell. The more careful taxonomic assignments going to have to come with Sasha's group and the DMARC program. But it looks as though it's a fully formed order springing into existence. And they tell us, I think, some interesting things about the origins of the original coronavirus, last common ancestor. The other group we have a lot of are Misoni-like viruses. And you can see they're branching all the way up the tree. That's the red things down toward the bottom. And we have some very early, quite basal branches on there. And with the longest branches, it's kind of an open question whether they fall just barely inside the Misoniviridae or whether they are sister groups to the Misoniviridae. And that's, again, a Sasha question hopefully we can answer. All right, here's the genome organization of the Proto-Nidovirales. And so landmarks, the pink thing running down the middle, that is our RNA-dependent RNA polymerase. It's picornavirus-like. Closest relative is the RDRP of coronaviruses. So, yeah, you know it's a relative. The purple right after that is your superfamily 1 helicase. And then you'll notice that polyprotein 1b just stops. No proofreading enzymes, no exo or endonucleases, no methylating enzymes down in that part, although you do get methyltransferases in other parts. The red boxes are a methyltransferase genetic signature. The other important thing to look at here, I think, is that structural gene organization down at the 3' end of the genome right-hand side. These are the stripy ones. You've got in every one of these a class 2 membrane fusion protein, which is going to be like alpha virus or flavivirus or bunyivirales. And it's overlapping with a nucleoprotein-like protein, which is basically the C-terminal half of a coronavirus-like nucleoprotein. And these things pop up in every single one. There's actually a lot less heterogeneity in this group than in the nitivirales proper, which is interesting. From these, we are able to spot a few additional landmarks. So these landmarks do not rise to the level of domains and are probably not very phylogenetically useful, but they are very useful when you get a new virus and you're trying to pencil in where the different regions start and end. So the first is the nitivirus helical landmark, nihil, and that one is a long alpha helical stretch that occurs after the main protease and NSP6, if this virus has one, before the NSP10 equivalent and the end of polyprotein 1A. And this thing is sometimes 500 amino acids contiguous that are all predicted by secondary structure predictors like JPRED to be alpha helical. Yeah, very striking. And the weird thing is that NSP8 of SARS coronavirus, this is our exemplar, it does form the exit tunnel on the polymerase, as you can see there in the polymerase reconstruction below. But it also has an enzymatic function, which is carried out by the blue beta strand domain shown over on the very edge. And that's the weird thing. That's the part that is not conserved. And if you'd asked me, I would have said, I predict the opposite would be true. But no, we've decided to keep the exit tunnel and lose the primase, basically. The other thing down at the bottom is there is something like an NSP10, meaning a small protein with a collection of 2 or more zinc finger-like motifs, so clusters of cysteine and histidine residues that are together. Now, these are involved in activating some of the methyltransferases, but they occur in many viruses where there are no functional methyltransferases to activate. So this is probably some kind of platform. It may join other proteins up to RNA. That's one of the features that NSP10 also has. I'd say the other thing to think about with those protonitivirales on the last slide is that they are missing the NSP8 primase. They're missing the NERAN nucleotidylating domain on the polymerase, and they're missing NSP9, where that nucleotide gets put to actually start the primer that the primase extends. So while they have the same sort of polymerase that's actually carrying out the rest of the synthesis, the starter mechanism appears to be completely non-conserved, and that is again a surprise. I would have expected that to be quite fundamental across the nitivirus. All right, next group. These are the Misoni-like viruses, and you can see a lot of these here. They're all from insects at the top, and down at the bottom we've got 2 that are actually quite large. One of these is 20,000, and the other one is over 35,000 nucleotides, complete genome. And Misoniviruses were named because they're kind of middle size between arterioviruses and coronaviruses. And yeah, I don't know. Maybe this is a lesson. Don't name your virus group based on its size because coronaviruses will surprise you. Or maybe these are branches, the long ones fall just barely outside the Misoniviridae proper. That is also possible, and that's something that will require Sasha and his analysis team to actually figure out. Otherwise, they are, pretty well-behaved citizens of the Misoni-like virus world. And almost all of the, all the viruses have a class 1 membrane fusion protein. So different from the class 2 that we see in the protonitivirales, but all of them also have that overlapping nucleoprotein, overlapping the N-terminal region of the glycoprotein. So the architecture's there, but they've swapped out a different spike. Yeah, so by the look of it. And the 2 that are associated with plants that we reported before also appear to have quite a bit of mitochondrial RNA from Tinaplusia, which is the moth on the left, and Franklinella, which is the thrips on the right. But We've been telling you about homology that you can demonstrate with programs like HHPred and HHBlitz, and those are the main tools that I like to rely on. They just find things that other homology detection software does not find. We actually have some homology down in the structural proteins, at least functional homology. Shown here on the left, these are the M proteins, probably homodimers in the membrane of several different coronaviruses. Shown here in the middle, these are the M and GP5 densities of arteriovirus. So this is the PERS virus from electron tomograms. And you can see it's pretty much looks the same. It's kind of doing the same thing. It's just a little shorter version of that. So M and GP5 are both 3-pass transmembrane proteins, and they have a about half transmembrane region at the front and about a half beta strand region at the back that may fold into a little beta barrel or beta propeller, perhaps. This is a similar architecture to what you see in the nidovirus, the other nidovirus M proteins, coronavirus M proteins. So it looks as though, even though these are outside the realms of what you can demonstrate with numbers, these are probably actually homologous, share a common ancestral origin. So the M proteins turn out to be vertebrate specific. You only find those 3-pass transmembranes with that architecture in the vertebrate viruses. And you find them in every single one of the vertebrate viruses that has been pulled out. So this is some kind of vertebrate adaptation, or an adaptation that happened to get into that branch of the tree and just never got out a single time. You see a mix of class 1 and class 2 proteins. The beta jelly rolls are in our out group, the caliciviruses. And every single one of these does have an N-like protein that is encoded. So something that starts with what is equivalent to the SR-rich region, which is kind of in the middle of the coronavirus N, and then appears to have something like the C-terminal domain of the nucleoprotein, which is the part that is conserved between coronavirus and arteriovirus. So it looks as though that is perhaps another universal nido and protonidovirus marker, but it may be too divergent to actually be phylogenetically useful, so we will see. Last virus, just to show you, these are the lito-like viruses, the early branches leading to the coronaviridae. And the only reason to put this one up is to show you that there's a bunch of neat fish and amphibians out there that have these similar viruses. But also we've managed to complete the N-terminal region of the polyprotein and actually the 5' region of the genomes of, I think, 5 of these now, including the microhyla virus that was partially incomplete before, and we've got the full thing now. So that's pretty neat. And yeah, the part where we had found it before, that is very sensible, looks very much like a coronavirus. Everything that comes before that in all of the viruses are quite divergent and interesting. The neat thing there is that that region contains the NSP3 equivalent, which is the big sort of prongs on that pore that comes out of the DMVs and presumably lets nucleotides in and RNA out. So, yes, anybody's guess. They may not have as much of a crown, or it may be a quite different looking structure there. It looks as though the pore part may be the conserved part and not the other part of that. Other conserved things that we find in these popping up again and again in these lineages. We've got eRNS. So this is one that you see in bovine viral diarrhea viruses. It's secreted RNAs that goes out and actually interferes with the function of white blood cells. We've got torsin 1A interacting protein 1 like proteins, but they only have the C-terminal domain. And you find that over and over, particularly in the litoviruses, but also in some of the new Misoniviruses. And interestingly, it looks as though this is only a partial gene, or rather a partial copy of a eukaryotic gene. And so this may be something like you see in the pox viruses, where they have decoys that'll have one part of the normal functional apparatus, and it'll just have nothing that it can signal to. So yeah, they may be decoy proteins. We've got replacing the papain-like deubiquitinating protein, 2 other kinds of deubiquitinating proteins. One of those that's a little more common is the OTU type deubiquitinating protein. They always occur in that N-terminal region, and so it appears that this type of protein in that type of region is quite generally and broadly used useful, but it doesn't matter which version you have. That can change on various branches. And you find the OTUs on the Protonidoviralis, so it's hard to say whether those are ancestral or the papain-like proteases are of the corona-like viruses are ancestral. You've got ALK-Bs and N6 adenosine methyltransferases, which we've heard some about during the conference. So these are a writing and erasing system for adding and removing methylation, which the cells will try to do to sort of slow down the virus, and the virus may try to do to some cellular genes to sort of slow down the host response. These quite often occur together in eukaryotes, and you get several of these viruses where they do occur back to back, probably as just one gene, which is both a writer and an eraser. So that's kind of interesting that so many of these viruses have picked that up. Last little bit of homology for you. This is the abyssovirus. So this is the one from the giant sea slug. And you can see here we've got DMV-like little vesicles, two membranes, little spidery stuff inside. That's probably the RNA. And we've caught a virus factory here as well. And so this is one that we predicted to be icosahedral enveloped with a class 2 fusion protein. And those are very consistently sized particles. It looks as though that may hold up, but we don't have quite enough information yet. But you can see some of those that look like little crescent moons that appear to be in the process of actually budding, so that's kind of interesting. And so overall, big picture, we can now look at a couple different regions. So the regions that do discontinuous RNA synthesis. Do we have evidence for the synthesis? And the answer is yes, in several groups, and in other groups we just don't have any evidence yet. So we need to learn, but we have actually pulled out subgenomic RNAs, many of them with leader body junctions, just like you would have in a real nitovirus, from many of these new viruses. So yeah, that looks like a pretty good conclusion, and a lot of the machinery that does that is intact. So I guess that makes sense. We see that the NERAN and the metal binding domain ahead of the helicase are not very well conserved. We're clustering these things as ancestral traits and derived traits, and this is based in large part on the position of the Protonidoviralis as ancestral to Nidoviralis and the place where the Protonidoviralis joins up to the Nidoviralis tree, which I think the best support for that is somewhere over in the invertebrate side. Let's see, in terms of the DMVs and their pores, the only part with any real conservation appears to be the NSP4 equivalent, transmembrane 2 region. The rest are kind of hit and miss. They come and go in different lineages. And we do not have full data yet on whether there are DMVs, but seeing it in something as divergent as an abyssovirus kind of suggests that this may well be what we see going forward. Lastly, virion architecture. Yeah, you have what appears to be a mix between class 1 and class 2 membrane fusion proteins, with the invertebrate viruses having almost all class 2 proteins. The vertebrate viruses, a mix of class 1 and whatever the heck arterioviruses have, whatever that is. But it usually is a set of genes that come in threes, and you'll have clusters of 3 or 3 plus 3. So the only real conserved structural feature there is going to be the C-terminal domain of the N protein and that highly basic, highly flexible region at the beginning, which is normally in the middle of corona-like viruses. The N-terminal domain only appears in corona-like viruses as far as we can tell from bioinformatics. So thanks very much. These are some of the people that helped immensely with putting the data for this together, and we're very grateful to our collaborators. And once again, time capsule, if anybody ever sees this out there in the future, I don't know, write and tell me, what's the future like? How are we doing? Did we get through COVID-19? I hope we did. Thanks very much, and all the best to you.